This video is going to be about the phases of mitosis. So the first phase of mitosis is going to be prophase. So during prophase, we're going to have our chromatins start to condense to form chromosomes, which we can see taking place right here. We're also going to have our nucleoli disappear. So if you look at this nucleus right here, there's no nucleoli, so it's already disappeared by this point. And then we have our mitotic spindle beginning to form, which we can see right here. So the mitotic spindle is a really important concept, so let's go and look at that a little bit more. So the mitotic spindle, a lot of events in mitosis are actually going to be very dependent upon the formation of the spindle, which, as we know, starts to form during prophase. So the mitotic spindle is made up of microtubules. So microtubules are made up of subunits called tubulin, and so to come up with enough tubulin to actually make this mitotic spindle, they do that through the depolymerization of the cytoskeleton. And so we know that the microtubules in the cytoskeleton, they can be depolymerized, so we'll take those apart and then use those building blocks to help make the mitotic spindle for this division process. So the mitotic spindle is going to originate from the centrosome, which we can see right here. And then it's going to attach to the kinetochore on each sister chromatid, which we can see as these black spots on these chromosomes. And so this attachment is really important because that's what's going to allow these sister chromatids to actually be pulled apart later on during this division process. So you might notice in this picture some of the spindle microtubules connect to kinetochores, but also some of them just connect to other microtubules. And so these are going to be our non-kinetochore microtubules, and so they will interact with each other, and they're going to play a really important role in actually helping this cell to elongate. That way it's going to be easier to divide and cleave in half later on in the division process. So the next phase is going to be prometaphase. So during prometaphase, we have the fragmentation of the nuclear envelope, which we can see taking place right here. We're going to have our chromosomes condense even further. So if you remember in the picture from prophase, these chromosomes were a bit larger, but now they've condensed even further. And then we're going to have our kinetochores form at the centromere of each chromatid. And so the mitotic spindle begins forming during prophase, but it won't really be completely formed until the end of prometaphase, and the kinetochores are able to attach to those microtubules. And so the microtubules begin to attach, like we just talked about. And then finally, we have the non-kinetochore microtubules interacting with those from the opposite pole, which we also touched on a little bit on the last slide about the mitotic spindle. So moving on to metaphase. So during metaphase, we're going to have the centrosomes relocate to opposite poles of the cell, because later on the chromosomes will get pulled toward these centrosomes because that's where the spindle attaches, so we want them to be on opposite sides of the cell. So the chromosomes are also going to line up at the metaphase plate, which we can see right here. And so this is just kind of the area where they all kind of meet up and hang out while they're waiting to actually be pulled apart. And then we have the kinetochores of the sister chromatids being connected to kinetochore microtubules from opposite poles. So for example, if we look at this chromosome right here in the middle, on this side, it's attached to the microtubule originating from this centrosome, and over here, it's attached to a microtubule originating from the opposite centrosome. So moving on to anaphase. So anaphase is when things actually get pulled apart and start to separate into two distinct cells. So anaphase, the first step is going to be our cohesin proteins get cleaved. So our cohesin proteins are found at the interface between these two sister chromatids, and so that's what's holding them together as a pair. So in anaphase, when we cleave those cohesin proteins, now those two chromatids have the capability to be pulled apart. And so the sister chromatids to separate, and they become chromosomes. So now they've separated, and each chromatid is now its own chromosome. So the two new chromosomes are then going to start moving towards opposite ends of the cell. So if you remember from the last slide, we talked about how the kinetochores on each of the sister chromatids are attached to microtubules from opposite poles of the cell. And so now those chromosomes are going to get pulled towards the centrosome that has the microtubule that is attached to their kinetochore. And so the cell is going to elongate as a non-kinetochore microtubules lengthen, which we also talked about a little bit when we looked at the mitotic spindle. So lastly, we are going to have telophase. So during telophase, we're going to have new nuclear envelopes forming. So now anaphase has separated these chromosomes from each other. And so now we can start to reform the nuclear envelopes to make it more like a normal cell. So we're going to have our nucleoli reappear, which we can see taking place right here. We're going to have our chromosomes unwind. So our chromosomes are going to start to relax as they move, as these cells will eventually move on into interphase and get ready for their next round of division. So our chromosomes are unwinding and our spindle microtubules get depolymerized. So in this picture, we can see that there's no more mitotic spindle because we don't need it anymore, and so we depolymerize it. That way we can use those subunits in the formation of the cytoskeleton, which a growing cell is going to be very dependent on. And so the last step of mitosis is the actual separation of these two cells. And so that process is called cytokinesis. So cytokinesis isn't really its own stage of mitosis. More so it starts at the end of telophase and then finishes up a little after telophase finishes. So what happens is we're going to form this cleavage furrow right here. And so this cleavage furrow is going to be made up of microfilaments. So that is going to be our actin and myosin, and it's going to form this contractile ring that is going to be able to cleave these daughter cells in two and actually separate them. So cytokinesis, like I said, is the division of cytoplasm, and it takes place beginning at the end of telophase. And so in animals, cytokinesis requires the formation of this cleavage furrow, which we've already talked about a little bit, is this ring of actin and myosin or microfilaments that will actually pinch the cell in two. And so yeah, the last part, just contractile ring of actin and myosin that allows for that contractile motion to take place. I hope you found this video really helpful. All images, unless otherwise stated, are from Campbell Biology's 11th edition. Remember that if you are an enrolled Baylor student, we do offer free tutoring on the first floor of the Sid Richardson building. You can schedule a free one-on-one 30-minute appointment, or you can drop in during any of our normal business hours. For more details, visit www.baylor.edu/tutoring.